So please have a seat. We are going to start. And this afternoon we have a session, Fungal Evolution in the Environment Reservoir. So it is the first time we use the word reservoir, as mentioned by Jacques. And we will start with Nicolas Papon. He is a professor in Angers University Hospital. In Angers University, sorry, and he's leading a research group in Angers. So, Nicolas, are you online with us?
Me.
Yes, Nicolas.
Okay.
Ah, on te voit. Yes, we can see you.
Yeah.
Perfect. So now we are waiting. We are waiting for the screen.
Okay.
Yes, I think it's good. So Nicolas is going to talk on fungal secondary metabolites and adaptation. Thank you very much, Nicolas. I know that it was not possible for you to be with us because Angers is not so far from Rennes, but thank you very much for this talk.
So, very much for the invitation. I'm very happy to contribute to the symposium. All of which is not here because, yes, I'm just from a on-site meeting in Angers June. I give you some basic information on metabolism in fungi. My pleasure. First of all, let me start by a short introduction on secondary metabolism within the tree of metabolites are a chemical compound with relatively complex structure. Here you got toxin produced by some coral. We know that toxin is not compound.
Nicolas? Yeah.
Yes.
Can you hear me? Yeah, there is a problem with your connection. It seems to be. So, can you? What do you think? You have to move your microphone.
Oh. Like this?
Lower.
Nicolas, if you put your microphone in front of your shirt instead of around your neck, the microphone will be lower.
Better like this?
Yeah,
oh.
Try it.
It's better like this?
Yeah, this is better.
Oh, okay. Let me a minute. This is on my second screen. I got the control of this.
C'est mieux, it's better. Yeah.
Better?
Yes.
Okay, so. It's okay for you?
Yes.
Yes? Okay.
Yes, it's okay for us. Thank you.
So, secondary metabolites are produced by winding metabolic pathway that are fueled by primary metabolism. The production of a down metabolite is usually clade specific, ranging from genus, species to strain. Concerning their general function, you must consider that secondary metabolites were evolved as a main chemical actor for interspecies defense, competition, and communication. For example, here, within the mint family, we know now that the biosynthesis of iridoids specifically emerged in the genus Nipita, yielding to the production of compounds that are attractant for phenins. And the fact that phenins turn around these grasses, this prevents too much attack by herbivorous insects. Surprisingly, we also know some examples where secondary metabolites mediate intraspecific communication. In flatworm, the depicted bat is specifically produced by the male to trigger the sexual maturation of the female and to promote the laying of eggs, which stands out, as you may know, as the main process, but condition the pathophysiology of schistosomiasis. In fact, secondary metabolites were described in near to all kingdoms within the tree of life, and their repertoire is mostly rich in plants, in bacteria, in fungi, and in a way also in animals. But for animals, this essentially involves aquatic organisms. Now, let me give you one example of prominent secondary metabolite per kingdom to illustrate their distribution within the tree of life. Here, maybe you know the Fungal alkaloids produced by some medicinal plants. These compounds such as vincristine, vinblastine are produced in leaf of these tropical species to defend against caterpillar attack. And these precious molecules are also known as first-line drug to treat cancer. A scoop on the right panel here, published in Nature 6 months ago, vinblastine is now produced in engineered yeast, in which researchers transfer in Saccharomyces cerevisiae origin, encoding dozens of enzymes for vinblastine heterologous biosynthesis. For illustrating bacterial secondary metabolism, I think you know the prominent example of ivermectin, because this compound emerged as the most potent antifilarial agent to fight elephantiasis in Africa, and this gave rise, of course, to a Nobel Prize in Medicine to you 8 years ago. And finally, for animals, let me introduce a paper in Science 3 years ago by Dalis Andes and colleague, reporting turbamycin as a new highly potent antifungal compound that is notably really active against the emerging yeast Candida auris. Okay, so now we will enter the core of the subject with a specific feature on the fungal secondary metabolism. Fungal secondary metabolite can be only detected in pluricellular fungi. I mean mold and mushroom, because genetic determinants for their biosynthesis are, yes, absentist. For instance, we know that 43 distinct secondary metabolites are produced by the mold Aspergillus fumigatus. As in bacteria, fungal secondary metabolites arise from complex biosynthetic routes that are directly fueled either by amino acid or acetyl-CoA from glycolysis. An important feature of these fungal biosynthetic routes is that gene encoding enzyme for a down compound here are organized in cluster. I mean, same region in chromosome, and in this cluster, notably in the center, we usually observe a core enzyme that defines the backbone of the compound, and around this core enzyme, we find a combination of decorating enzyme gene. In the evolution of fungi, gene clusters are mostly ones circulating by vertical transfer, and in a lesser extent by convergent evolution and horizontal gene transfer. The main families of fungal secondary metabolites include terpenoids, polyketed non-ribosomal peptides, some hybrid molecules, and some other less frequent types. And first, terpenoid. The biosynthesis of terpenoid is driven by coenzyme named terpene synthesis. Here on the right panel, a scoop in the field recently published in Nature, discovering in fungi a new path for terpenoid biosynthesis thanks to a new class of bifunctional terpene synthase. Fungal terpenoids include flavor that perfume, for instance, aged cheese. And terpenoids in fungi also include trichotesines that are the main part of the dangerous exposome, notably responsible for unsanitary house syndromes. Miroterpenoids are also a specific subfamily of terpenoid well known in fungi such as the fumagillin, produced by Aspergillus fumigatus, and used to treat microsporidia infection in bees. So let's go for the second prominent family of fungal secondary metabolite, I mean polyketids. Polyketids, the backbone of these compounds, are produced by multimodular enzyme referred to as polyketid synthase. Sterimatocystin is one of the most famous fungal pro-inflammatory polyketid, and for instance, we have recently shown in our laboratory that Scelosporium, which is the main mold that colonizes the lung of patients with cystic fibrosis, downregulate the biosynthesis of this sterimatocystin to evade the host immune system and to promote a long-term colonization in this patient. Most of the so-called mycotoxins also belong to the polyketid family in fungi. They are highly toxic for cattle and human health, and they are produced by several molds that develop directly in grain stock used for feeding livestock. And as polyketid, at the last polyketid example, we can cite lovastatin, which is produced by some mold and as a powerful inhibitor of a specific enzyme of the metabolic pathway. It blocks in nature the biosynthesis of essential steroids in microbial competitor. And as you may know, statins are also in human health a great drug to treat high blood cholesterol. Now the third family, the NRPs. The backbones of NRPs are produced also by multimodular enzyme synthase. The first example, of course, is penicillin, historically identified by Fleming in the modern Penicillium chrysogenome, and you see here the historical petri dish that allowed the identification of these famous antibiotics. The other excellent example to illustrate the family of NRPs is cyclosporin A, and this compound, as you may know, remains one of the most used anti-calcineurin immunosuppressant in solid organ transplant patients. As the main example, we can cite for hybrid molecules. We can cite, of course, and introduce echinocandins, such as the caspofungin, which is a semi-synthetic natural product initially extracted from the Ascomycete glarea lozoiensis and now widely used to treat deep-seated candidiasis. And finally, some others anecdotal type of fungal secondary metabolites include, for instance, indoalkaloids produced by the rye ergot and well known for their pharmaceutical properties. And a last example of secondary metabolite of this subtype is a trimorgenic mycotoxin, which disrupts the GABAergic amino acid neurotransmission, and this induces tremoring when you ingest contaminated food. So now, what about the functions known for secondary metabolite in fungi? Basically, secondary metabolites are involved in fungal development. For example, if you knock out the PKS4 gene in Soldaria macrospora, you will lose the capacity to produce perithecea. And if you, on the right, overexpress this gene, you will observe a huge fruiting body in this trait. We also know for a while that pigment, such as melanin, protects fungi from damaging UV radiation. Secondary metabolites are, above all, broadly used by fungal species as defense chemical and weapons against microbial competitors to infect plants and also to fight insects, for instance. It's important to keep in mind that all experiments in the literature investigating the antimicrobial activity of extract are finely biased. Why? In terms of concentration, if you compare to real ecological concentration, and it is highly likely that physiologically relevant concentration of a specific secondary metabolite could function only as a signal rather than a weapon in the nature. The proof that secondary metabolites are defense and weapons in fungi in nature is very easy to observe in your laboratory. For instance, if you treat Aspergillus foliatus with echinocandin, so a natural product, you will detect many changes in the regulation of signal transduction pathway, leading to overexpression of gene encoding enzyme involved in many different biosynthetic pathways for secondary metabolite in response to this input signal. In most of the fungi, iron acquisition is also mediated by a secondary metabolite. For instance, in our lab, we recently demonstrated that in Scedosporium, once again, in the number of patients with cystic fibrosis, these more produce NRP type siderophore that scavenge iron from Pseudomonas pyoverde. Another interesting question in the field is how fungal producers of toxin can prevent self-intoxication, because of course most of the time the produce secondary metabolite as a direct target in situ in the fungal producer. So for instance, here in pink, in Fusaria, that produce the T2 toxin, well known, a specific pump is encoded in cluster and mediate the efflux of the toxic compound outside of the cells. In some other cases, the fungal producer expresses a detoxifying enzyme that converts the toxin to a non-toxic derivative. This is here the case of gliotoxin, for instance, produced by Aspergillus fumigatus. Other possibility, many more species use gene duplication for amplifying the target and prevent self-intoxication. This is okay, or some more that produce statins. And sometimes this gene amplification is also coupled with the selection of mutation in the target sequence for promoting the resistance, because this second isoform is less susceptible to the toxin. Important fact here, this means that browsing fungal genome for this type of resistance gene is one of the most powerful strategy for drug discovery. And finally, 2 slides concerning future development in drug discovery. During the last decade, secondary metabolite discovery has been mainly supported by the explosion in the number of fungal genome available, and coupled with a powerful online software. With this recently led to the identification of hundreds of new fungal biosynthetic gene clusters. Once a new gene cluster is identified, we benefit now from terrific advance from the synthetic biology field for characterizing, but also for producing at high scale the corresponding natural product, notably by engineering 2 main cell platforms, chassis, Saccharomyces cerevisiae, of course, and also as a more modern Aspergillus nidulans. So As a conclusion, we can remind that only few hundred gene clusters are characterized to date, and on the basis that molds usually are more 30 to 70 gene cluster per species, and that to date 20,000 molds are described. There are more than 1 million of clusters to be discovered in the future, and this is obviously an underestimation. Anyway, I hope I convince you here that fungal secondary metabolism will continue to boom as an excited field of basic and applied research for years. I thank you very much for your attention.
Thank you very much, Nicolas. So the connection was good. No problem.
Okay, thank you.
And thank you for your fantastic talk. Metabolites, secondary metabolites are less known than other topics, so I think it is really important to have this conference within a session on evolution and on adaptation. So, thank you very much. Is there one question? Otherwise, it is just the time for Johanna Rhodes after. So, no. Okay, thank you very much, Nicolas. And we move to the next talk. So, Johanna Rhodes, thank you very much for being here. You are working in the lab of Matt Fisher, and we are very happy to have you regarding your topic on evolution of Aspergillus. Thank you. Genomic evolution.
Okay. Brilliant. So we're going to be diving down into the genetics. We have some fantastic talks, and thanks to Nicolas for his talk as well, which I will actually touch upon in mine. So I'm working on an antifungal resistance project with Matt Fisher, but I'm also based in Paul Verbee's lab in Radboud UMC in the Netherlands, and he will be giving a talk in the next session. Next, here we go. So just disclosure, funding from Wellcome Trust, and I've also received an honorarium from the Gilead Sciences. So Aspergillus as a genus is really varied. It's found in a diverse set of environments, both terrestrial and also aquatic as well. We're thinking about Aspergillus sedowi, which causes this sort of infection in fan corals and just basically eats them away. And then, of course, we also know Aspergillus infections in humans, and then also in plants as well. So it's kind of a really sort of interesting set of organisms that can just cause so many opportunistic infections, such a wide variety of environments. And it's an efficient saprotrope. And as you can see, it looks phenotypically quite varied. So let's have a look at some of these different Aspergillus species. So Aspergillus flavus, I kind of like this one. It kind of looks like if you've cut a kiwi in half. And that's what it reminds me of, but I wouldn't lick it, I wouldn't eat it, that's for sure. It's opportunistic, and it's a destructive agricultural pest that contaminates crops with what's called aflatoxin, which Nicolas has just sort of hinted on. And it causes crop yield losses, but it can also enter the food, the consumption, food cycle, and cause human death in rare cases, but also hepatocellular carcinoma. And it's estimated there could be anywhere between 25,000 and 155,000 cases worldwide each year attributed to aflatoxin ingestion. I'm sure we all know about Aspergillus fumigatus, listed as a critical priority fungal pathogen on the WHO list that came out last autumn. There's high rates of azole antifungal drug resistance infection, and this varies depending on the country because of different surveillance techniques and programs, so anything between 47 and 88%. And it causes invasive infections, particularly invasive aspergillosis. Aspergillus niger is widely used in the food industry for enzyme production and can sometimes be the cause of some cases of pneumonia. And again, like similar to flavus, it can secrete these toxins, this mycotoxin called ochratoxin, which causes problems with the kidneys if ingested and hazardous to human health. And the last one I wanted to touch on was Aspergillus nigulans. So this has really been kind of like the model organism. And Aspergillus fumigatus has kind of taken over in terms of genetics, but Aspergillus nigulans was one of the first that people were really looking at in terms of genetics and genomics. And it's considered an important model for eukaryotic genetics and cell biology. And one of the echinocandin drugs is actually derived from a fermentation product of a variety of this as well. But what is in a name? So where does Aspergillus, the name, actually come from? It's actually kind of interesting. So it's first described about 300 years ago by a priest and botanist, Antonio Micheli. And Aspergillus actually refers to the asexual life cycle. So fungi can actually undergo asexual life cycles when the conditions aren't really favorable, and it will actually produce clones of itself. But they also have the ability to reproduce sexually, produce more diversity because they have mating types. So basically, the phenotypic diversity of sexual fruiting bodies is greater, and different genera describe the resexual cycle. So for instance, Aspergillus fumigatus is the asexual, whereas Neosartoria fumigata is the sexual. And this paper that came out a decade ago, can you believe, it just shows the variety, the diversity of Aspergillus as a genus, using Penicillium chrysogenum and Marneffii as outgroups. And this is just basically using one reference genome per species, just to give you an idea. And in the column, the first column, you might not be able to make out the numbers, but these are the genome sizes, and then the next column is the associated gene content. And if you can just about make it out, the genome sizes range from about 28 to 40 million base pairs, and the gene content is anything from about 9,000 to 14,000. And what's neat about Aspergillus is that they all seem to arrange into 8 chromosomes and a mitochondria. There might be some sort of deviations from that, but generally speaking, that's what they do. And there's actually 250,000 genomes publicly available for the Aspergillus genus. That's more than Cryptococcus and Candida. It's not quite as much as Saccharomyces cerevisiae, which has over 1 million, but it's still a lot. So actually, the Aspergillus genus is well placed to be a model genus for our genomics and genetics investigations for eukaryotics in general, but also Aspergillus and fungi. So let's get into the nitty-gritty, genomics. And some of you might actually be too young to remember this, but most of us probably do. Less than a quarter of a century ago, the human genome was published, and it cost $95 million US. Now, thankfully, it's a lot cheaper. It's only $1,000-ish to sequence the human genome. And with the falling cost in sequencing a whole genome, for humans, it also falls for fungi as well, so we can take advantage of that. So we have sequenced more fungi because it's more accessible and because it's cheaper. And the beauty of genomics is that we can use it for a range of things, but there's certain things that we need to take into consideration first. Firstly, the DNA extraction method. So if we want to do short read sequencing, so Illumina, you need to do a certain extraction method. If you want to do a long read using nanopore, MinION, or PacBio, you need to do a slightly different extraction method. And it also matters when you're doing your extraction. So as I mentioned, there are sexual life cycles, asexual life cycles. When are you going to take your DNA extraction? Should you extract from the spore or the hyphae or the fruiting body? And there's all these things. And the majority of the time, actually, the extraction is just done whatever is there on the lab bench at the time, which perhaps isn't so great. So we could actually see different results in our sequence data depending on when we do the extraction. But the beauty of the genetic data is that we can do genome assemblies and structure studies to look at actually what is in these genomes, functional annotation, pathogenic microbiology to look at what are the resistance mechanisms that are conferring drug resistance, and then the comparative genomic analysis within and then also between species. So there's remarkable genomic sequence diversity within Aspergillus. So there's a few more fungal species included here, but Aspergillus as a genus is kind of just this bit here. And for the majority of the isolates included here, there's just one genome, but a few of them actually have some more. So there's 2 genomes, 4 genomes for Fusarium. And then when we get into Aspergillus, we're actually starting to see a few more being sequenced, but mainly just one representative genome there. So it's actually as diverse as our own phylum, the vertebrates, but the close relatives Fumigatus and Fisheri are as divergent as humans and mice. So there's a heck of a lot of diversity within the genus. And although the genomes are quite stable, generally ranging from that 28 to 40 million base pairs, they are not what we would call plastic genomes. I know that some people do think that the Aspergillus genomes are plastic. They're not as plastic as other fungal genomes. And they do have several similar characteristics. But there are a lot of differences. And we think that these differences, well, we know that there are differences because of this. It's because of horizontal gene transfer. And that's what these arrows are symbolizing here. So the big arrow here symbolizes a big horizontal gene transfer event from Aspergillus nigulans into Podospora, and then other various events occurring. So now that genome sequencing has become more accessible, we can sequence more genomes than ever before. And there's these really big genome data sets available and being published. So actually, it is worthwhile doing pan-genome analysis. This is where we can take all of the isolates within a species or a clade, and we can look at what genes are common to all of them, and then what are not common, i.e. accessory. So we're just using this little diagram I've created here. So the yellow gene and the blue gene would be what's called core, because they're present in all 3 isolates. The purple and the pink genes would be in the accessory genome, and this little turquoise gene here would also be in the accessory genome, but it'd be called a unique gene because it's only found in one isolate. So with this analysis, we can actually reveal more diversity. So when we look at the Aspergillus genus as a whole, it actually, the pangenome shows that there's way more diversity than we thought. And there's just under 63,000 gene families across 36 Aspergillus species. And only 6% are found in all genomes. That's incredible. So this diagram here, we've got the gene count in the red, fairly similar across the species. Same for the core genome, and it's the species-specific genes where we see the diversity. Anything from 182 for Aspergillus niger, all the way up to 3,000 here for Aspergillus ellipticus. So we can actually see quite a range, and that means if we sequence only 1 or 2 or 3, just a handful per species, we're missing out on so much diversity. What we've also seen with pangenome analysis is that there's evidence of gene loss, duplication, and extensive horizontal gene transfer. And we think that this is what is driving the diversity within this genus. So let's have a look at Aspergillus fumigatus specifically, because this is where a lot of the research has been carried out. The Aspergillus reference genome was created about 30 years ago. There are multiple, but there's two that's sort of the main ones. And they generally agree in that they contain about 10,000 genes over 8 chromosomes and a mitochondria that equals about 29 million base pairs. And then over the last sort of 18 months, there's been 3 studies looking at pangenome analysis, and it's revealed way more diversity than we thought. And what was really nice is that these 3 studies all included the same isolates as a subset of their pangenome analysis, and then they added additional ones for their studies, perhaps specifically in a geographical region, or ones that have collected perhaps a little bit more clinical or more environmental. So as you can see, the mean genome sizes are roughly the same for the 3 studies, but we actually found quite significant differences. So Lotus Lofgren found the most. She found 15,000 genes for her pangenome. In our study, we found about 13,000 genes, and Amelia Barber found just under 11,000 genes. So actually, we can find massive differences. And it's important to note that the mean genome size is the mean genome size. So that means that the range is something else. And I can't speak for Lotus or Amelia, but what we are seeing with our range of genome sizes, some of the Aspergillus fumigatus genomes are up to 70 million base pairs, and the gene content is off the chart. It's new stuff that we'd never seen before, and it's really quite exciting. We wouldn't have found that if we haven't done pan-genome analysis. And what we all agree on in these 3 studies is that the core genome was enriched for housekeeping functions, but the exciting stuff is in the accessory genome. And this really, really does link in quite nicely with the previous talk. Secondary metabolism is a big part. And secondary metabolism is a significant factor for the delineation of Aspergillus as a whole, but it's also linked to azole antifungal drug resistance. So let's just touch on secondary metabolism in Aspergillus as a genus. Yes, it's commonly unique to individual species, and there's a higher number of secondary metabolism gene clusters in Aspergillus than there is in Penicillium. So there's just under 3,000 in over 37 Aspergillus species. And the dynamics of secondary metabolism suggests that horizontal gene transfer, so we're coming back to that horizontal gene transfer, is basically the mechanism of what's driving the evolution here. And this is really apparent because if you look at this phylogeny here, you can see that there are quite some distinct Aspergillus species. So for instance, Flavus is down here, and it's very different, say, to the Aspergillus species up here. There's one secondary metabolism gene cluster that was found in 5 really distinct species, and the only way it could be present is through horizontal gene transfer. When we looked at secondary metabolism in Aspergillus fumigatus, it was really quite interesting because we looked at what loci in the genome were approaching fixation. And this means, basically, is the loci found in all the isolates in that population. And if they were, we would expect to see it a dot right at the top of the plot. And what was interesting is that we saw, for this FST, we saw like these waves across the genome. And these high regions of FST mirrored significant gene phenotype associations from a GWAS study, which linked loci to itraconazole drug resistance. And these regions, these peaks and these shoulders, these significant loci were actually involved in secondary metabolism. So basically, what we think is happening is horizontal gene transfer is basically literally moving segments of the genome, conferring resistance to azole antifungal drugs, and then also they are linked to secondary metabolism as well. We took 28 gene deletion mutants from the COFUN knockout collection and screened them for growth on media containing itraconazole. And we saw that there were 2 isolates that were not able to grow, one that had CYP51A knocked out and one that had ABCA knocked out. And I'll come back to that in a minute. So what is actually driving the evolution? We've got a how, got horizontal gene transfer as the how, but what is driving it? So it's selection pressure, which is basically external agents that are just basically causing this change. They could cause a negative change, so for the trait to basically die out, or positive change that causes the increase in the trait. And positive doesn't mean it's positive for us, it means it's positive for Aspergillus. And it's just basically this whole mismatch of stuff. We've got change in agrochemical use, change in at-risk groups, change in climate, change in biota, change in virulence, and then also the way that we as humans use resources. So we're spraying chemicals environmentally in agriculture, but also in horticulture, animal husbandry. We're composting, which is actually a significant factor as well, but we're also using the same chemicals in agriculture that we're using in the clinic as well. And all of this together is creating pressure. In Aspergillus fumigatus, drug resistance mechanisms are primarily centered on CYP51A, and that was the gene that was knocked out in the isolate that couldn't grow on itraconazole, which is what we'd expect to see. And it has a multitude of resistance mechanisms that are pretty unique for Aspergillus. And the structural polymorphisms, tandem repeats of sequences in the promoter region, the most prominent being a tandem repeat of 34 base pairs. This causes overexpression. and then also duplication events. And we see drug-resistant Aspergillus fumigatus in the environment at high levels. We would expect to see it quite high in agricultural settings, so say for like arable farming, but actually it's only about 1%. We see more drug resistance in urban flower beds, and that's at 14%. And we also see a heck of a lot of drug resistance in air samples, and this does follow a seasonality pattern. We are more likely to find drug-resistant Aspergillus fumigatus in the spring than, say, summer and winter. There we go. And this mirrors clinically as well. So if we're finding it in the soil, we're finding it in the air, then we as people are breathing it in. And azole resistance was present in 13% of isolates from cystic fibrosis patients, and about 27% of them had this tandem repeat 34 mechanism as well. And what's really interesting is that these azole-resistant isolates have a fitness advantage over drug-susceptible isolates. So there's a different mechanism occurring here in terms of microevolution. So these isolates have advantage in acidic pH and also high histamine and histidine conditions, which are the conditions that are primarily found in the cystic fibrosis lung. So we're seeing not only selection pressure from external to us, but from internal, adaptation to the human host driving evolution. By sequencing a heck of a lot of isolates, over 1,000, we can use a population genomics approach to see that azole-resistant Aspergillus fumigatus has spread globally and increased in prevalence over time. So that TR34 drug resistance mechanism is here in blue. And what we can see is that the majority of the isolates are actually in this bottom section of the phylogeny. And then the azole-susceptible isolates are in the top part of the phylogeny. When we map this same phylogeny onto resistance to agricultural fungicides, we see that the pattern is the same. The majority of the resistance mechanisms are in this bottom part of the phylogeny, and the susceptible ones are in the top. And what's interesting is this contains a mixture of both clinical and environmental isolates. So these clinical isolates, they're only going to acquire isolates that are resistant to environmental fungicides by breathing them in. They are not going to be treated with an environmental fungicide. So patients are acquiring infections from the environment, and these infections are pan-drug class resistant. The pangenome for the Aspergillus fumigatus actually revealed additional information. So what we saw was that there were some drug-susceptible isolates on a different genetic background. They were in that group at the top of the phylogeny where we'd expect to see susceptible isolates. And they didn't contain any known mutations in CYP51A. And what the pangenome analysis revealed was that there were genes in the accessory genome that basically point to alternative resistance mechanisms associated with secondary metabolism that are driving a different mechanism for resistance. So resistance, in fumigatus at least, is definitely more complex than we thought. But what about mechanisms in other Aspergilli? So this is a paper by Emil Gluck-Thaler. He's currently in Neuchâtel, and he's moving over to America this fall to take up a tenure-track position. And he's done extensive work on what's called starships. He's a massive Star Trek fan, so that's why they've been called that. And he's done this analysis over multiple fungi, but he's found these starships in Aspergillus flavus. And starships are these huge mobile elements that facilitate variation in gene content, and they basically just pick up and move. They're like transposable elements. They are about 110 kb long on average, and they share conserved components and carry diverse arrays of accessory genes. And they're a newly discovered route of evolution that can be transmitted not only through horizontal gene transfer, but also vertical transmission as well. Other mechanisms include transposable elements and repeat-induced point or RIP mutations. So I'm thinking to the work of Clutterbuck, who sadly passed away last autumn. But his work in RIP mutations in fungi is unparalleled and also really hard to understand. It's quite dense, but it is good. And then we have seen the activation of transposable elements in stress conditions in various Aspergillus species, and that the transposable elements are involved in the regulation, again, of secondary metabolism. It keeps cropping up, which means it must be important in Aspergillus nidulans. So to summarize, we've learned that sequencing one genome per species is insufficient because it really doesn't give us the depth of knowledge about diversity, and we miss a heck of a lot of gene content. Pangenome and population genomics analysis offers way more opportunity to identify diversity, and diversity associated with drug resistance as well. Aspergillus genomes are not plastic, but they are evolving, and this is because of pressure from us as humans and the Anthropocene activity. Azole resistance in Aspergillus fumigatus, at least, is linked to secondary metabolism. And on the same background as the azole resistance, we're also seeing this resistance to agricultural fungicides as well. And we can also see adaptation of fumigatus to certain patient cohorts in this microevolution at like the loci level in cystic fibrosis, but also in other respiratory conditions. And I didn't mention it, but we don't see it in COVID in kappa patients. Whether that changes remains to be seen, but I doubt it would. There are various pressures driving evolution in fumigatus, and fumigatus is responding in many ways, and we're not even keeping up. We are woefully falling behind. But what can we expect from other Aspergillus species? There's a bit of a sort of like a disjointed way that we approach analysis in the Aspergillus genus as a whole. There seems to be a certain level of analyses that continue in fumigatus and a different sort of analyses that occur in the other Aspergillus species. And they're finding out different things. It'd be really good if we could kind of work together and mesh them together and see what we can find out. Thank you so much for your attention. This work would not be possible without all of these people, and I hopefully have given you a nice summary of where we are with the state of genomics and Aspergillus.
So thank you so much, Johanna, for this brilliant talk. Very nice slides. Do you have a question for Johanna, even if it's very...
I appreciate it was very diving in, so no question is stupid.
Particularly on the genomic of Aspergillus. So Jacques, yes, you want? Yeah. Thank you.
Thank you very much for the very nice talk. I read with great interest your recent paper about the outbreak of aspergillosis in non-flying parrots.
Oh,
so
yeah.
As a vet, I'm very
yeah,
I
that
like
was many long nights.
Very nice story.
Yeah.
I had a question about the potential microevolution regarding the host. So, for example, in birds. So, any information?
Yeah, so it's a bit of an odd one because the Aspergillus fumigatus outbreak in the kakapo was actually quite different to what we would see in the human host, really. So there was some sort of genetic distinction there. And whether that is representative of Aspergillus infections in animals in general remains to be seen. I've got 3 genomes to look at from a dolphin and a cow type thing and a moose to look at and see whether they are sort of more related compared to humans. But yeah, so it was a very distinct genotype. Yeah, but in terms of microevolution, it seemed to evolve less than what I would see in humans as well. So whether that's because there's less pressure, maybe, I don't know, or it's just a very different background and it doesn't have the same mutation rate, doesn't have the same clock. Basically, I'm kind of more inclined to think it has a different clock. Yeah, but the jury's out on that one. We'll see. Ask me in a year's time.
Thank you.
Yeah.
Okay, thank you. Yes, one more question.
Joe, thanks for great talks. I have a question because you just mentioned the pangenomic probably will be future direction, bioanalyze genomic data. But if you want to find out transmission route, for example, you can link the source and on patients, so is the pangenomics will help?
maybe to an extent. You have to remember that Aspergillus fumigata, well, Aspergillus in general, has a lot of recombination. So how do you define a cutoff as to what is a similar isolate in terms of transmission? Transmission software and methods and algorithms that are out there at the moment rely heavily on a clonal genome, which is why it works so nicely on bacterial pathogens and then Candida auris as well, which is kind of the outlier. For Aspergillus, it might be incredibly tricky, and you will probably end up applying those methods or having a very arbitrary cutoff that doesn't really make a lot of sense. So would the pangenome help? Potentially, if you kind of said, okay, well, the core gene content is high, but the accessory gene content is low between these isolates. Whereas if they were very disparate and there was no transmission, you might expect more accessory gene content. But that wouldn't be a clear-cut answer, I don't think. Yeah.
Okay, so it was a great pleasure to listen
Thank you.
to you, Johanna. Thank you very much. And we move to the proteomic approach after the genomic approach with Vishu from Pasteur Institute in Paris. He's heading the Aspergillus unit. So, Vishu, the floor is yours.
Yes. Hello, everyone. First of all, thank you, Jean-Pierre, for inviting me to be a part of this symposium. Yeah. So, I'm a basic researcher. I don't touch any patients. I touch only mice. Okay. So you see a lot of data regarding basic research in my presentation. So the topic what I'm going to talk about is a proteomic to study evolution of Aspergillus. So I have nothing to disclose. So when you look into proteomics, you can look into two different aspects. What happens to the fungal side when fungus changes its morphology from dormancy to germination. But this is a bit confusing. As you see here, if you use a different medium, RPMI, Brian, minimal, Zapac, and collagen, the way you separate protein is completely different. So it is chaotic. So it's very confusing. But still, we are working with this proteomic analysis, how the proteomics changes from dormancy to germination. So we are working with the two different fungal species, Aspergillus fumigatus and flavus. Probably this is not a good context to speak about today here. So I moved to proteomics from the host perspectives. So what happens when fungus is there in normal or healthy human, and when the infection starts? So, you know, today's talk will be mostly focused on Aspergillus fumigatus because that is the focus of my lab, but we also work with Aspergillus flavus. So you all know, owing to the small size and hydrophobicity of spores, they can reach up to the level of lung alveoli. So if the spore is reaching the macrophage, there will be phagocytosis, there will be cytokine production, and there is recruitment of immune cells to the site of infection. Then finally, fungus will be cleared in a normal condition. But what happens if it is going somewhere else, if it is not reaching the macrophage? So then it has to face alveolar fluid. So we had the question, what it interacts with. So for that, we did a very simple experiment. We took a BAL sample, bronchoalveolar lavage, from control individuals. So here I say control, it is not healthy individuals because it's a collection of BAL is a invasive protocol. So we collected BAL from control where there was no fungal, bacterial, or viral infection. So we opsonized conidia. We took out spores, conidia, and we extracted what is bound on the surface. We did the proteomics. So this is the long list of proteins what we saw. So most of them are complement proteins. So here you see two types of extraction. One is hydroxylamine extraction, and the other one is sodium thiocyanate. So here, this extraction will remove those which are covalently bound, whereas here it is non-covalently bound. So we have to focus on this one because there is direct, strong interaction between spore and the proteins. So, as the major components found were complement proteins, we had the question what it leads to. So, what we saw, when there is unopsonized spores, they are all dispersed. When there is opsonization, they become aggregated. So, whether it has any implication on phagocytosis, yes. If you see unopsonized spores, they are taken up very less compared to opsonized spores here, what you see here. So, as most of the proteins which popped up were complement proteins, we had the question whether there is any implication of complement receptors. So, what happens when there is opsonization by complement? It will be taken up by complement receptors, because this is important here. There is a lot of emphasis on dectin-1. Dectin-1 is the receptor which phagocytoses fungal spores. But here, what we saw was when we blocked complement receptors CR3 or CR4, what we saw was there was significant decrease in phagocytosis. So, there was 60-65% decrease in phagocytosis when we blocked complement receptors. But I would like to warn here, even unopsonized spores are taken up. That means complement receptors are not only the receptors involved in phagocytosis. So, we did another set of experiment. So, the normal route of inhalation is through intranasal route. So, when we took wild type and C3 knockout mice, C3 is the central molecule in complement activation. So, what we saw was there was no difference between wild type and C3 knockout mice. On the other hand, if you inject spores through intravenous route, of course, we saw difference in survival. So, this is the survival curve. So, we used different concentrations or different numbers of spores. As you see in all different MOI, there was a difference in wild type and complement C3 knockout mice. So, what we saw was there was decrease in phagocytosis of spores in C3 knockout mice, both by neutrophils and monocytes compared to wild type mice. So, when we saw complement system is implicated in phagocytosis and clearance, we had the question, what is the difference if we opsonize spores with serum and BAL? Because in vitro system, most of the time we use serum, not BAL, because it's difficult to get BAL. You clearly see here, TNF alpha was more produced, IL-6 was more produced, or IL-8 was more produced if you opsonize spore with serum. On the other hand, if you opsonize spores with BAL, there is more production of IL-1 beta. So, there is a pathway difference if you opsonize spores with either spores or BAL. So, this led us to question, is there any difference between the opsonization pattern? Of course, we saw difference in the opsonization. So, some proteins were only present, only binding to spores when you opsonize with serum, or some proteins were only present when you opsonize spores with BAL. So, one of the major or interesting point here, interesting protein was surfactant protein D. That's because surfactant protein D is the major humoral immune component present in the lung alveoli. The main function of surfactants is to prevent the collapse of the lung alveoli during respiration, but surfactant protein D is also a C-type lectin. It is having lectin property. It has pattern recognition receptor property. So this is the structure. It has a carbohydrate recognition domain. There is a collagenous region, and we saw that surfactant protein D is directly interacting with spores. We proved it by immunolabeling as well as by FACS analysis. So, what is the implication of opsonization by surfactant protein D? So, it was increasing phagocytosis and it was increasing ROS production. So, before Nicolas was saying, if there is polyketide, it tries to inhibit or it tries to reduce the production of ROS, but when spore is covered by melanin, but over that, if there is SPD binding, it increases the production of ROS and increases the clearance of spores. So we proved it by animal study also. We used wild type or SPD knockout mice. As you see here, most of the cytokines were less produced in case of knockout mice. So that was about spores, but we also had the question what it does if fungus is already germinating. So if you cultivate spores in the presence or absence, you see there is clear difference in the germination rate. So there was 60% reduction in spore germination in presence of surfactant protein D. So this is the morphological defect. Control, it is nicely producing hyphae, but if you add SPD, there is defect in the surface of hyphae. So here I would like to point 30 micrograms per ml. The physiological concentration of SPD is 50 to 90 micrograms in BAL. So we are much below the level of physiological concentration. So it was this binding or alteration of the surface of hyphae was also affecting the cytokine production. It was causing immunomodulation. Most importantly, if you add SPD to uriconazole combination treatment, you see increased efficacy of antifungal drug. So that was mainly because if you use control, this is chalcofluor white staining, which binds to the cell wall chitin. So here, control, it was not going inside, but if you treat with SPD, chalcofluor white is going inside. That means there is pore formation in hyphal cell wall if you treat it with SPD. On the other hand, that was about healthy condition. We looked at infectious condition. So what we saw was, this is the volcano plot. There was upregulated and downregulated proteins. So there were 8 humoral immune factors completely absent in infected condition. So here we took the BAL from 10 patients, 4 were colonized, 3 were IPA, and 3 were CPA. So interestingly, SPD, C1q, Ficolin 2, and MASP2, CFHR 2 and 5, MRC, and C8 are the proteins which are completely absent. On the other hand, in the infectious condition, pentraxin 3 was highly present. So the question here now is whether we can use these humoral immune factors as diagnostic markers. That is one possibility. So we also had the question why pentraxin 3 is highly present here. That's because pentraxin 3 is not binding to dormant spores directly. It binds to swollen spore or germinating spore. So probably there is no interacting partner here. On the other hand, when we opsonized first spore with SPD, then we added pentraxin 3, there was increased binding. That means the binding partner is missing in infectious condition. So here, SPD alone, when it is opsonized, it is producing high level of cytokine. But when SPD over that, if you add pentraxin 3, there is lower amount of cytokine production. So probably this is a kind of regulatory mechanism because initially there is SPD. So SPD is binding to spores. There is pro-inflammatory immune reaction. On the other hand, once it is produced, then there is the production of pentraxin 3. It binds to SPD. Then there is less production of cytokine. It's a kind of tolerance mechanism. So there are a lot of studies on correlation with the infection and all these humoral immune factors. For example, pentraxin 3 SNP, single nucleotide polymorphism, is known to be high risk factor for invasive aspergillosis, same like MASP, that's a serine protease. On the other hand, in mouse model, it's already been shown that SPD knockout mice or C1q, FCN2, pentraxin 3 knockout mice are highly susceptible to invasive aspergillosis. So the question is whether we can use these components for therapeutic therapy of IPA. For example, it's already been shown in mice model. If you add SPD intranasally or pentraxin 3 intravenously, there is increased survival of IPA mice. So that leads to the question whether they are having any therapeutic benefits. Most interestingly, what we did was we took this mutation, this resistant mutant, and here you see it's resistant to voriconazole, but when you add SPD, it doesn't matter. It behaves like reference sensitive isolate. So that means SPD is also active against azole-resistant isolates. So now we have done RNA-seq analysis to see what's the mechanism of action. So there was a lot of emphasis on secondary metabolite. What we see is that SPD is shutting down all secondary metabolite formation in Aspergillus fumigatus. So it is not only shutting down secondary metabolites, it is also decreasing many more pathways. So probably we are going to present that data in AAM next year. And just today morning I got data from Netherlands. It shows that there is a single nucleotide polymorphism in SPD, which makes patients more susceptible to invasive aspergillosis. So that's about the host side. So proteomic from fungal aspect. So here we try to extract surface proteins from dormant spores, and during germination, we saw that there was overlapping protein in both conditions. But interestingly, what happens is if you give this extract to the neutrophils, then there is less production of IL-8 or less phagocytosis. So that means fungus is trying to counteract the defense mechanism of the host system. So we know, I think today morning there was a talk about metalloprotease and alkaline protease. So what we found was there is metalloprotease and alkaline protease associated with fumigators, which are trying to counteract this humoral immune factors. So altogether, host is trying to kill fungus, but fungus is also trying to counteract the host system. So with that, I hope that proteomic, I convinced you that proteomic provides a platform to understand host fungal interplay, because we see the reality here, because I'm not against genetics and genomics, but genetics and genomics, we don't see the reality, because, for example, there are 9,900 genes, but when you see proteins, you see only 5,000 proteins. So it gives the question whether we have to simultaneously look into proteomics or not. So with that, I'm ending my talk, and these are my acknowledgements. Thank you.
Thank you very much, Vishu, and thank you to raise the debate between genomic and proteomic. Johanna, do you have something to answer?
Because I think altogether now only 5,000 proteins have been identified out of 9,900 genes. Yes. So where is the rest? No, I'm not saying it is one is important, one is not important. I think parallelly one has to look into.
Yeah, as I said, they are different sides of the same coin. But it is a really good point. Is what is happening with the other 4,000 or more? You know, if we see more gene content in other fumigators, what is happening to those? Are they redundant? Are they junk?
That's right.
What is it? And so on and so forth. But I really enjoyed that. And what was interesting was that SPD shuts down secondary metabolism in Aspergillus. Did I get that correctly?
Yes. So that's what we are observing. It is diminishing or down-regulating all the transcripts of secondary metabolites.
Interesting. That's really fascinating. I just thought it was brilliant. Thank you. Yeah.
Thank you. Yes, Rina. Yeah. We have time.
I found that fascinating from at least one point of view. It's been years I played with complement, and I love somebody getting back to complement work. Yes, it's massively important and it has a central role. And I feel so uncomfortable when we look at in vitro models where complement is ignored because it's complicated and it's difficult to add.
Right.
Your serum gets inactivated and, what do you do? And your bugs die and you just leave it out. And then you do massively fine proteomics or other analyses on a sort of half a model. I really like that it's done properly. But same applies to the genomic and looking at the gene expression side of things that, you have to have the whole model there to get anything. Just like you have to have the right phase and the right side of the fungus itself.
Yeah. That's what I'm a bit worried about some of the research. I'm not commenting on anything. For example, most of the in vitro studies have been done using fetal calf serum, which is far away from human serum. Okay, so it's depleted it is heat inactivated fetal calf serum what we use, wherein you shut down all complement system. And it's low in immunoglobulin. So we are ignoring many things there. So,
and the other side of the story is that we've learned in the past already that, of course, human body protects itself from its own complement by massive array of regulators. And some microbes can exploit those regulators and use them for their own protection. Candida known for that, for factor H and that sort of stuff. So I think it's really important to realize that the bigger picture is far more complicated.
That's true. And we look at so many chemical compounds. Again, I'm not against antifungal drugs. Antifungal drugs are targeting only one enzyme, whereas if you look at SPD, I have not given you the complete picture. It is downregulating all siderophore forming proteins. So siderophore is important for iron acquisition for fungus. So for fungus to germinate, iron is essential. Okay, so it is attacking multiple pathways. So first, I think we have to look into what is the normal condition to combat any fungal or any microbial pathogen. Then probably it will give or lead you to a better idea how to tackle it with.
And so, Vishu, do you think that in the near future we will have to screen our patients for the SPD polymorphism in order to evaluate their susceptibility to Aspergillus?
That will be very fantastic because that's what I got the mail this morning at 11. There is one SNP particularly with a probability value of 0.05, which is quite interesting, showing the risk factor for IPA. So
perhaps, yeah. But 10 years ago, we had the nice paper from Vishu, I don't remember his, and collaborators, and they showed that TLR4 polymorphism could be associated to the susceptibility in allogenic stem transplant patients for Aspergillus. But 10 years after, we don't do that.
So TLR is a receptor which is expressed depending on the presence or absence of any microbial threat, whereas SPD is constituently present in the lung because it's the purpose of SPD is something structural. So probably it is more stronger candidate to look for than all TLR or any other components, because TLR is it's highly expressed or low expressed depending on the presence or whether there is any microbe or not. So that same thing holds good for cytokines also, because I know some people are interested in cytokines, but what time you will be looking at cytokines?
And you mentioned susceptibility to invasive aspergillosis, but regarding chronic aspergillosis? or ABPA? What do you think?
We see that because the proteomic analysis, what we did was with a combination of colonization, CPA, and IPA. So it is absent. That means it is absent in CPA condition also. So ABPA, I don't know.
Okay, thank you very much. We have 5 minutes, and I learned during the last meeting in WHO that now we can. It's nice to have 1 minute stretching, moving from our chairs, etc. So if you want, you can stretch and do what you want during 1 or 2 minutes. You can upload, you can do everything. But now, yeah. In a different international meeting, I saw that. So it's very. Oui, il y avait, c'était des APC de chez moi. Ouais. Mais bon, en fait, il n'y en avait pas beaucoup, donc ça ne permet pas de. Non. Yeah. So just stretch and not moving from. You can stay here. We are going to move to the next presentation. C'est différent de se lever et puis de sortir.
You made such a rich program.
Yeah.
It has to be 5 days, not 2 days.
Yeah, that's right. But nice talk. So, our speaker is moving, so. Ouais. Yeah. So, no, it's not a break. Something. No, just, 1 minute to stretch, but they moved. So.
Sorry, they all went to the toilet, so...
Yeah, okay, we are going to... But we were 5 minutes early. So.
Oh, wow.
Yeah, we are very good for that. Ça va, Sandrine? Ouais, très bien. Bah, c'est super, mais là, c'est dommage parce qu'il y avait plein de propos sur l'évolution génétique, protéomique, et tout ça. Rhodes qui a fait un topo magnifique. Vishu, le master, là.
Mais je voulais arriver
je te propose... Enfin, là, il y a juste un topo, mais de te mettre devant, il y a des places devant, si tu veux. Mais sinon, il y en a à l'arrière. Okay, so we are going to move to the next talk. So please, we have the coffee break in half an hour.
Je sais pas si ça marche.
C'est celui-ci. Hein.
Non, mais il m'a dit pour le pointeur.
Ah, d'accord.
Mais il n'y a pas de pointeur.
Mais sur les écrans, les pointeurs, c'est pas très très bon. Mais bon.
Non. Donc là, tu avances comme ça.
Ouais.
Plus next. D'accord.
Ouais, voilà. So. Now it's my pleasure to introduce Marie-Elisabeth Bougnoux from Pasteur Institute and Necker Hospital. She's a medical mycologist and doing very nice research on Candida in the team of Christophe Denfer, Pasteur Institute. Thank you, Marie-Elisabeth.
Thank you, Jean-Pierre, and thank you, Jean-Pierre, for the kind invitation in this wonderful meeting here in Rennes. And thank you for giving me the opportunity to talk about genome evolution of Candida. And I am going to focus my talk today on a specific question that is how the evolution of Candida genomes enable the acquisition of phenotypes important for Candida pathogenicity. Among all Candida species responsible for infection, Candida albicans is probably the most relevant species for deciphering this question, because Candida albicans is a diploid, has a diploid genome with a complex reproduction, and also because Candida albicans evolves all their life cycle in an unique environment, the human being. Actually, no environmental source, no terrestrial life size have been observed. Candida albicans is a commensal list of oral, genital, and intestinal tracts. And recently, we show that Candida albicans colonize almost all healthy individuals, but with very low quantity and with very variable quantity. Candida albicans, you know, is also a major opportunistic pathogen responsible for superficial infection, but also for invasive infection in immunocompromised patients, and also it's associated with high mortality. Therefore, in human, Candida albicans is confronted with very different environment, different niches with stressful environment which require significant genetic adaptation. This is a short reminder that the genome of Candida albicans is diploid and with a pair of chromosomes, chromosome 1 to chromosome 7 and chromosome R. But it is very important to keep in mind that the genome of Candida albicans has a high level of heterozygosy with an average of one heterozygous position every 200 base pair. In comparison, in human, there is one heterozygous position only every 2 kilo base pair. There are a lot of mechanisms driving genome changes from nucleotide to whole chromosome levels, and the genome of Candida albicans is able to tolerate rearrangement such as aneuploidy and loss of heterozygosy, LOH. And these LOH events can cover short regions or large segmental part of chromosome or even whole chromosome. And just to remember, using data from whole genome sequencing of multiple clinical isolate, we demonstrated and we confirmed that the population structure of Candida albicans is organized in different genetic clusters, gathering strengths genetically very close, and also we confirm that the reproduction is predominantly clonal with diversification with SNPs and LOH. We showed also that we can see gene flow events between genetic clusters, but it is a rare situation. Now I would like to just focus my talk on a specific cluster, the cluster number 13, because strands of the cluster are phenotypically different of strands of the other cluster. These strands are less fit, less virulent. The strand of cluster 13 were called Candida africana because the first isolate were from Africana. And these strands have a high capacity to colonize genetic tract and cause vaginal infection. And genetically, these strands are highly similar with reduced heterozygosy. well, it is very well, it's very well possible to see in this figure, because you can see the density of heterozygosity along Candida albicans according to the cluster. And if we compare the density of SNF, we can see that for cluster 13, the color is blue, because the number of heterozygosity is very low. And we demonstrated finally that some genes important for virulence are disrupted in cluster 12, and these trends are very important in the virulence, and can explain why we have a cluster 13 restriction. We found an accumulation of homozygous codon non-stop due to nonsense mutations, specifically in cluster 13, and this codon non-stop are fixed in this cluster, in this clade. Therefore, here you have an interesting and unique example of niche restriction in Candida population due to extensive LOH with accumulation of deleterious mutation in genes involved in the fitness of Candida albicans. But during infections, infectious strains can also undergo different gene variations, and we know that LOH, the frequency of LOH events increase in response to different stress, such as antifungal exposition. And this was very well demonstrated by Alix Coste several years ago, and she thought that after acquisition of mutations in ERG11, gene encoding for azole target, or TAC1, encoding for transcription factor, with the regulation of efflux pumps, you obtain and you observe the full advantage of this mutation when the strength is in homozygous state following LOH events. Therefore, LOH is a major driver of Candida phenotypic adaptations. Therefore, and we just learned that aneuploidy also can facilitate adaptation to niches. This is a new paper that showed that when strains of Candida albicans were placed in gastrointestinal tract, we can see the emergence of strains with a third chromosome 7. Actually, these strains with a third chromosome 7, with the trisomy of chromosome 7, are more fit in competitive experimentation in gastrointestinal colonization. But these strains have a low capacity to produce filamentous cells and it is a very different phenotype. Actually, on the chromosome 7 is located NRG1. NRG1 is a gene that encodes a negative regulator of filamentations, and it was demonstrated by the author that NRG1 inhibits filamentation of Candida albicans in a gene dosage dependent fashion. Therefore, when a strand carried third chromosome 7, these strands have a fitness advantage in gastrointestinal colonization model. This is the first demonstration that the possibility that we can have a reversible adaptation to different host niche by gain or loss of aneuploidy. And during infection, genetic change can occur in infection strands, and often these genetic change are major. And I would like, for illustration, to present a short case report of relapse of infections in immunocompromised patients. In these patients have had candidemia, and with blood, catheter, and urine culture positive for Candida albicans, and he was treated by fluconazole, and 14 months later, the patient has a relapse with a multifocal arthritis with culture positive for Candida albicans from hip, ankle, and knee. And if you look at the whole genome sequences, and if you compare the whole genome sequences of these three strands collected at the same time, you can see different evolution of the genome of each strain. For example, you can see an increase of copy number variation with in hip strands the presence of aneuploidy in chromosome R, and also in chromosome R you are a trisomy, and trisomy chromosome 1 in the strands from ankle. Secondly, you can see large LOH tract events in the chromosome 2 of the hip strands and in the chromosome R, on the chromosome R of the strand from knee. And interestingly, this case shows that each strands have evolved independently each other. And it's very interesting. And if you look at the summary of all genomic alteration can occur over time and between the different infectious seeds, you can see that each strand has developed a specific rearrangement. And what are the significance of this difference and this rearrangement in terms of pathogenicity? What are the significance of this rearrangement for the relapse of the infection? It is not possible to precise. We will add some other experiments to better understand the role, the impact of this rearrangement in the tissue and in the pathogenicity of the infection. In conclusion, the main mechanism responsible for evolution of genome during both commensalism and infection are LOH and aneuploidies. LOH is frequent in Candida albicans, with evidence for active diversification of Candida albicans through short-range LOH during commensalism, evidence of a large range LOH during infection in human, and evidence also that LOH can be associated with fitness advantage. And recently, we learned that the major influence of aneuploidy on Candida albicans pathogenicity through changing in the expressions of virulence factors and the interaction with the host. What next? It's your volunteer. I think that the future longitudinal study should assess the evolution during infection of the genes changes to better understand the real contribution of these rearrangements in the pathogenicity of the candidiasis. Just to finish, I would like to thank the unit at Institut Pasteur, Biology and Pathogenicity unit, and specifically Corinne Maufray, Margot Delavie, Jeanne Ropars, Natacha Sertour et Christophe Denfer, that people working specifically in this STEM, in the unit. Thank you for your attention. Do you have some question about this evolution of Candida albicans?
I always do.
Thank you.
I can keep you entertained. Sorry. Yeah, my question is about polyploidy.
Yes.
And we have a patient who has become colonized, has a CMC syndrome, has become colonized with the panazole-resistant Candida albicans, and we've used one drug after another, latest one, ibrexafungerp. And with every drug, now we've been recycling them for her management. And she responds to all of the drugs for 2 weeks and then just stops. Whatever drug you throw at her, ambisome, ibrexa, and the only explanation that comes to mind is that there is something like polyploidy happening, and it's just the fungus is producing so much target protein or something, and then when we grow it on the plate for a couple of generations, it doesn't look like it's resistant to anything. Because it goes to euploidy, probably. We haven't tested that, but it's just single patient. But could it happen that quickly? And would that, what do you think, what's going on?
Yes, it's all the question, because we don't know exactly the significance of these rearrangements, but these rearrangements finally are very frequent. And it's possible that these rearrangements allow the persistence and the relapse of the infection. For example, is a multifocal arthritis. Is not usual in during candidiasis. These patients has polyarthritis, and it's possible that after the strength is located in the hip, and in the hip several rearrangements occurred, and after we have another infection. And it's possible that this rearrangement, and these strands carrying this rearrangement are lost. And it's very important to understand, to better understand the contribution of this rearrangement, because it's probably very important for stop the infection and to, why not, to find prophylactic antifungal or prophylactic.
Yeah, in her case, she has a syndrome. She will always get oral and other Candida infections over and over again. So we just have to play with it.
Yes.
Okay, thank you very much. Yes, one more question. So after the debate on proteomic and genomic, Aspergillus and Candida, what is the most interesting?
Hi, thanks for your great talk. So I'm just wondering, how stable is aneuploidy? What do you think? How stable?
Aneuploidy is not stable.
Yeah, but then is it?
And it's very important thing,
Yeah.
because it's things important in the fact of the adaptation is reversible, because you can gain aneuploidy, and you have an adaptation because you have a third chromosome and genes with activity, with different expressions, and we can lose this aneuploidy. And it's, I think that is very important. It's very major driver of evolution, fast, rapid evolution during infection in human. In human.
Yeah, if it's unstable, how
in animals.
If it's unstable, how can you catch this aneuploidy and you get the good sequence?
Yes.
If it's unstable, so when you culture it and you do the extraction, when you sequence them, and
yes.
how can you easily catch the aneuploid stage?
Yes, but I catch I catch aneuploidy, but the aneuploidy is stable in the culture. Yes, in the culture. Yes, it's stable. Yes, in the culture. But if you pass these strands in animal models, in the gut of animal model, you can lose the aneuploidy.
Okay, thanks.
Yeah, because I think that's exactly the question. That let's say that the Candida has 4 chromosomes or 3.
8.
8.
Yes.
No, sorry, I didn't mean the chromosomes, but copies of each,
copies.
being polyploid.
Yes.
And that has happened because of the antifungal pressure in the patient. You take the fungus out of the patient, grow it happily on sab agar with no antifungals, just nice food. Why would it carry on with that polyploidy? And how quickly does it go back? Because why would it bother?
Yes, probably.
To be polyploid.
Yes.
So it probably goes back to euploidy. So exactly if I wanted to show that my patient's isolate does this switching from euploidy to polyploidy and back and forth, how can I show it? Because genetically, it just looks like there is 1.5 fungal cell. How do you measure it? How do you show it?
Yes, we will measure this frequency.
An idea about that is, technically it is unstable, and how can you catch it? Aneuploidy happens for a reason, and we kind of say, oh well, it's due to pressure, to antifungal drug stress, but what triggers the aneuploidy, and we don't know. So it's that you've got to look for. What is the mechanism that causes the ploidy to essentially turn on? That's how you know whether your patient's got it or not, but we just don't know what it is.
Yeah, so that would be pre-growing the isolate, or when taking the isolate from the patient, having a bit of azole in the plate and keeping it sort of teased. Patient, yeah.
I wonder if you could do the reverse, put some antifungal drugs on your culture to try if you get the the polyploidy.
Polyploidy, yeah.
Yes.
And how long it takes.
But how do I measure it then? How do I show it's happening?
You will answer for us.
Okay, so thank you very much, Marie-Elisabeth, and now we have a coffee break. And thank you for this excellent session on evolution. Johanna, Vishu, Marie-Elisabeth, and Nicolas in Angers.